My name is Danika, and I will be presenting the role of the non-neuronal cholinergic system in immune regulation. To begin, the immune system is essential for the host's defense against potentially invasive and dangerous pathogens. Inflammation is the immune system's response to harmful stimuli, which may include things like pathogens, damaged cells, toxic compounds, or irradiation. Its goal is to remove the injurious stimuli and initiate the healing process. Once the immune system detects the harmful stimuli, signaling cascades occur. So, immune cells like monocytes and macrophages release inflammatory mediators, as well as plasma fluid containing proteins like fibrin and antibodies that help with the healing process. Furthermore, small messenger proteins called cytokines are released, which initiates immune signaling and inflammatory responses. Cytokines may either be pro-inflammatory, meaning they promote inflammation, or they may be anti-inflammatory, meaning they resolve inflammation. Some examples of pro-inflammatory and anti-inflammatory cytokines are listed here. Inflammation may also be acute, meaning it only lasts a short period of time and is typically not problematic. It can also be chronic, in which the inflammation lasts weeks and even months, which may lead to tissue damage and autoimmune diseases. These immune responses are essential to combat various pathogen and harmful stimuli, but when the immune system becomes unregulated, for example, if cytokines are being uncontrollably released from immune cells, it can result in autoimmune diseases like multiple sclerosis and rheumatoid arthritis. In MS, the immune system mistakenly attacks the myelin surrounding the nerve fiber, causing miscommunication between the brain and the body. In rheumatoid arthritis, the immune system attacks body tissues like cartilage, causing pain and damage. As I previously mentioned, the immune system consists of various immune cells like monocytes and macrophages, and their dysregulation may contribute to inflammatory and autoimmune diseases. Although I will be discussing particularly monocytes and macrophages in this talk, these cells are not the only cells that make up the immune system and that participate in such diseases. In this study, I am more interested in studying macrophages. Macrophages are differentiated monocytes, and they exist in a spectrum between M1 and M2 macrophages, M1 being pro-inflammatory and M2 being anti-inflammatory. It is important to note that most macrophages exist in a combination of phenotypes, and these extreme polarities are uncommon. Another important aspect of the immune system is the cholinergic system. The cholinergic system plays important roles in immune regulation. The cholinergic system refers to cells capable of producing the required elements for acetylcholine synthesis, storage, transport, and degradation. These elements include choline acetyltransferase, also known as CHAT, acetylcholine esterase and butyrylcholine esterase, also known as ACHE and BCHE respectively, and vesicular acetylcholine transporter, also known as VACHT. Although acetylcholine is typically viewed as a neurotransmitter, it plays important roles in non-neuronal cells, commonly known as the non-neuronal cholinergic system. Acetylcholine is not only a neurotransmitter, but it can be released by and act on non-neuronal cells like immune cells. Of particular interest in this study is the role of acetylcholine in immune cells. Studies have shown different immune cells capable of producing or degrading acetylcholine. Extracellular acetylcholine produced by some of these cells can then act on a variety of different nicotinic acetylcholine receptors. Nicotinic acetylcholine receptors are pentameric ligand-gated ion channels with important roles in not only immunity, but also in signaling, learning, memory, and reward pathways. These receptors have recently shown to be involved in immunity. More specifically, the alpha-7 homomeric receptor is of particular interest in this study. It is the most abundant subtype, and activation by agonists like acetylcholine and nicotine can protect against inflammatory and autoimmune diseases, mainly by inhibiting the cytokine production and therefore reducing the overall immune responses. Typical agonists like acetylcholine and nicotine are effective in reducing cytokine regulation. However, they are not specific to immune cells. Therefore, other molecules produced by doctors in Florida have shown to be able to bind to the alpha-7 homomeric receptor and reduce cytokine release. These molecules are known as silent agonists. silent agonists are DEPP-based analogs. Underneath each of these molecules are their scientific names, and in parentheses are the names that we use in our labs, and they'll therefore be referred to this combination of letters and numbers throughout this presentation. These novel drugs induce a prolonged desensitization of nicotinic acetylcholine receptors with minimal channel opening, resulting in an overall anti-inflammatory effect. So the objective of this study is to gain a better understanding of the anti-inflammatory properties of these silent agonists in human macrophages. It is hypothesized that the silent agonists will decrease immune responses by inhibiting the release of pro-inflammatory cytokines. For this study, the THP1 cell line was used. This is a human monocytic cell line in which I differentiate them into M0 or M1 macrophages. To differentiate both types of macrophages, PMA was used, but to further differentiate into M1 macrophages, interferon gamma and LPS was also added to the culture medium. These conditions were chosen based on previous literature and experiments showing to induce M1 differentiation. These are pictures of my cultured cells and show some morphological differences between undifferentiated M0 and M1 macrophages. I started off by first identifying cholinergic markers in THP1 cells via Western blot. The first lane here is a positive control, and the next two lanes are my THP1 protein samples. As you can see, there are two bands for chat, one around the predicted molecular weight of 82 kilodalton, which is more pronounced in the gel where the bottom bands are covered. And the second band is around the 40 kilodalton, and this is verified by a different chat antibody. This can result from either a slice variant or a different isoform of chat. Then, the presence of BCHE, VCHT, and ACHE were analyzed again by Western blot. Again here, the first lane is our positive control, and the next lanes are the THP1 samples. Here, we do not see the presence of BCHE or ACHE in our protein samples. They can only be seen in the positive controls. However, we do see VCHT in both our positive and our protein sample. These results confirm the presence of some important cholinergic markers. Next, I conducted a PCR to confirm the presence of the alpha-7 homomeric nicotinic acetylcholine receptor within my THP1 cells. I did not perform Western blot to identify the presence of this receptor like I did for other cholinergic markers, simply because there are no good antibodies to identify these specific receptors. As shown in this agarose gel, we see a band very close to the predicted 335 base pair amplification, confirming the presence of the alpha-7 receptor within the THP1 cells. Now I'll go into the CBA results for the M0 and M1 differentiated cells. Here we have different conditions. So we have a DMSO alone treated group, an LPS plus DMSO, and then the LPS plus the silent agonists. The reason why we have a DMSO control is because our silent agonists are diluted in DMSO, so this acts as the vehicle control. LPS stands for lipopolysaccharide, and it's found on gram-negative bacteria. It's used to induce an immune response within our cells. So now jumping into the results, we do not see any significant inhibition in both IL-1 beta and IL-6 release when treated with the silent agonists. Here I show TNF-alpha and IL-10 again for the M0 macrophages. 6MQ39 and 2MQ103 seem to inhibit TNF-alpha, but this was not shown to be statistically significant. For the IL-10 release, 6MQ39 has a significant impact on IL-10 release, reducing its release from 100% to 79%. Again, in this graph, it seems as though 2MQ103 has an effect on IL-10 release, but this was not shown to be statistically significant. These results therefore show that 6MQ39 and 2MQ103 have potential to reduce both TNF-alpha and IL-10 release in M0 macrophages, which may be involved in the progression of various autoimmune diseases. Now onto the M1 cytokine results. Here we see a significant inhibition of IL-1 beta when treated with 2MQ75. No other significant inhibition is seen in this graph, and no significant inhibition is seen in the IL-6 release when treated with the silent agonists. IL-1 beta is often involved in the progression of chronic inflammation, therefore reducing IL-1 beta release by treatment with 2MQ75 may have some beneficial outcomes. And finally, here we have TNF-alpha and IL-10 release again in M1 macrophages. We see a significant decrease in TNF-alpha release from 100% to almost half, so to 47%. 2MQ103 also seems to have an influence on TNF-alpha release, but this inhibition was not shown to be significant. For IL-10, no significant inhibition is seen when treated with the silent agonists. Considering the involvement of TNF-alpha in various autoimmune diseases and chronic inflammation, these results show that 6MQ39 has great anti-inflammatory potential. I would like to thank my supervisor, Dr. Alain Simard, at the Northern Ontario School of Medicine for his guidance and all of his help in the lab. I would also like to thank all my lab members, Dr. Ramya Naranjula and Dr. Natalie Lafort for all their support and help in the lab. Finally, I would like to thank Dr. Hornstein and Dr. Papke from the University of Florida for the synthesis of the silent agonists used in this study.